Hello, my name is Nicole Verdun, and I'm a Super Office Director in the Office of Therapeutic Products in the Center for Biologics Evaluation and Research here at FDA. And today I'm here to talk to you about innovative therapeutics, gene therapy. The mission of the Center for Biologics Evaluation and Research is to ensure the safety, purity, potency, and effectiveness of biological products, including vaccines, allergenics, blood and blood products, cells, tissues, and gene therapies for the prevention, diagnosis, and treatment of human diseases, conditions, or injury. Through CBER's mission, the center also seeks to protect the public against the threats of emerging infectious diseases and bioterrorism. The Super Office of Therapeutic Products was established in March of 2023 in the Center for Biologics Evaluation and Research, with the goal of creating flexibility and capacity for future growth, and to improve discipline alignment, increase review capacity, and enhance expertise in both cell and gene therapy. The Super Office of Therapeutic Products is made up of 6 offices: the Office of Gene Therapy, Chemistry, Manufacturing, and Control, or CMC, the Office of Cellular Therapy and Human Tissue, CMC, Office of Plasma Protein Therapeutics, CMC, the Office of Clinical Evaluation, the Office of Pharmacology Toxicology, and the Office of Review Management and Regulatory Review. OTP regulates a diverse array of products, including gene therapies. The gene therapies that we regulate include ex vivo genetically modified cells, non-viral vectors such as plasmids, replication deficient viral vectors such as adenovirus, adeno-associated virus, or lentivirus, replication competent viral vectors such as measles, adenovirus, and vaccinia, and microbial vectors such as listeria and salmonella. In addition, we regulate stem cell and stem cell derived products, terminally differentiated cell therapies, therapeutic vaccines, and other antigen-specific active immunotherapies, such as cancer vaccines and immunotherapies such as dendritic cells, lymphocyte-based therapies, cancer cell-based therapies, peptides, proteins, and non-infectious disease therapeutic vaccines, such as peptides, proteins, and small molecules. In addition, we regulate blood and plasma-derived products, such as purified and recombinant proteins for hematology or coagulation factors, xenotransplantation, and in the past year, we have approved both a pig heart and kidney for transplantation, human tissues, and also some devices. Now I'm going to talk a little bit about gene therapy product development. Gene therapies can be delivered via either ex vivo or in vivo administration. With in vivo administration, on the right side, the gene therapy product is administered directly to the subject. As an example, here you can see an adeno-associated virus vector or lipid nanoparticle encapsulating nucleic acid, such as messenger RNA. These vectors could be administered intravenously, where they could travel to the liver perhaps and correct a monogenic disorder. In ex vivo administration, cells are removed from the patient and genetically modified in culture outside the body with vectors such as lentivirus or naked DNA. The genetically modified cells are then reintroduced into the patient. One example of this is CAR T cells for cancer therapies. There is a diverse array of gene therapy products. The figure here shows the 2022 database of the different vectors used in gene therapy clinical trials. A large portion includes replication deficient viral vectors, such as adenovirus, AAV, retroviruses, and lentiviral vectors. Other vectors include non-viral vectors, such as plasmid DNA, mRNA. In addition, you have ex vivo genetically modified cells and replication competent viruses and microbial vectors. Interactions with us occur at various stages of product development, starting with interact meetings. which could be used for early discussion on CMC, pharm tox, or clinical related issues. Interact stands for Initial Targeted Engagement for Regulatory Advice on CBER or CDER products. And you can, at the time of these Interact requests, have preliminary evidence or proof of concept studies and really want to engage with the FDA early. In addition, you see we have several different phases of meetings, including at the pre-IND phase, end of phase meetings, pre-BLA meetings, mid-cycle and late cycle meetings. In addition, we have an entire set of meetings that you can request that are discipline specific, and we welcome all communication with sponsors. Key differences in gene therapy development are, unlike other medical products, gene therapies often involve new technologies to manufacture the product, sometimes invasive procedures to administer the treatment, potential long-lasting effects, which makes it different than drugs, and potential risk of unintended changes in DNA or rejection by the body. So all of these are careful regulatory considerations when we are discussing products with companies and with academia. Considerations for chemistry, manufacturing, and control of gene therapy products in development can be quite complex. In particular, in early phases, it's important to have characterization of your product, understand the mechanism of action as much as possible. Considerations of the cellular kinetics and vector biodistribution are also needed. And in later phases, quality and development of potency assays is quite important, but it is equally important to consider these things in early phases of your product lifecycle. Major manufacturing changes can also require demonstration of product comparability. Prolonged biological activity and the need for long-term follow-up are particular considerations for gene therapy products. And the procedures for product delivery, including product device compatibility, monitoring for immunogenicity during development, and monitoring for off-target effects, including tumorigenicity and insertional mutagenesis. Particular considerations for preclinical development include the development of animal models in both healthy and disease-related states, depending on the product. And scientific justification for model selection is needed. In addition, comparative physiology and tissue type helps with extrapolation to clinical dose levels. Product kinetic profiles, such as the cell fate or vector biodistribution. The route of administration should be as close as possible to the clinical scenario in the preclinical development phase. The timing and rate of administration, anatomical location, activity of the product, and the local microenvironment are all considerations in the development of your preclinical program. In addition, standard toxicology assessments such as mortality, laboratory observations on treatment, body weights, gross and histopathology, and other endpoints as recommended in the current guidance documents and ICH guidelines. And an informative design with randomized group assignments in animals, appropriate controls, masked assessments, adequate study duration, and the evaluation of the product's persistence are needed. Some of the regulatory considerations for clinical development in early phase include dosing considerations. So the preclinical data is used to inform or support dosing used in early phase trials, and looking at potential toxicity, which will be informed through some of the preclinical data. In addition, what is needed is a scientific rationale for dose escalation or de-escalation, adequate characterization of the safety profile of feasible doses, and considerations around delivery devices or the route of administration. For safety, we often look at dose-limiting toxicities, the duration of follow-up, ensuring that it is tailored to individual products. Are there staggering regimens and stopping criteria in place for both individual subjects or the entire study? And to consider monitoring for immunogenicity, evaluations for viral vector shedding if needed, And is there an acceptable safety for the population in the context of this disease or the condition that's being studied? In addition, we have to evaluate for product persistence and think about long-term effects, even in early phase trials. For efficacy considerations, the approval of drugs and biologics must be based on substantial evidence of effectiveness and evidence of safety. And available data has to demonstrate that product's benefits outweigh its risk at the time of approval and throughout the product's life cycle. Certain aspects of product development that are feasible for common diseases may not be feasible for rare diseases. And examples of regulatory flexibility include single-arm studies to support regulatory decisions. In situations where we have a large magnitude of benefit, we often have some flexibility in terms of the framework for the efficacy studies. And well-designed natural history studies can be supportive for rapidly progressing, serious, and rare conditions. We also have to give consideration to any associated companion diagnostics or devices that are co-developed to become available for use with the particular product. And we use expedited programs and other incentives for serious diseases with unmet medical needs. Some of the expedited development programs are listed here on the slide and include Fast Track, Breakthrough Therapy, RMAT designation, which is a program specific for regenerative medicine and advanced therapies in the Center for Biologics, accelerated approval, and priority review. To highlight RMAT, because it is specific to CBER, what is needed is preliminary clinical evidence that indicates that the drug or biologic has the potential to address unmet medical needs for a disease or condition. Now we will talk about further considerations for investigational and approved gene therapies. Gene therapies hold great promise, and over 80% of rare diseases have a known genetic basis. So far, FDA has approved 22 gene therapy products, most of which are for rare disorders. OTP currently oversees more than 2,600 active investigational products, and collaboration and flexibilities are really critical in the development of cell and gene therapy products. This slide just illustrates the tremendous growth that we have seen in the gene therapy space in recent years. Here you see the number of gene therapies that have been approved, and as you see, it also reflects the tremendous growth in the past 5 years. So this is quite a new and evolving field, but it is also quite exciting as we have several more therapies that are in the pipeline for development that truly address unmet needs. This slide highlights some of the recent cell and gene therapy approvals from the Office of Therapeutic Products, and it really does highlight the diverse array of cell and gene therapy products that are being developed. It's quite exciting. In particular, here we have a product from Orchard Therapeutics that was approved in March of 2024 for metachromatic leukodystrophy, a product from Pfizer that was approved in April, which is an AAV vector used for the treatment of moderate to severe hemophilia B, Adaptimmune's product for treatment of unresectable or metastatic synovial sarcoma. We have Autolus's product for the treatment of adults with relapsed or refractory B-cell precursor ALL, and PTC Therapeutics has a product, a gene therapy for the treatment of adult and pediatric patients with AADC deficiency, which is the first product approved for this condition. One of CBER's 2024 priorities, and a priority for the FDA in general, is to further the development of products for rare diseases and for diseases where there is significant unmet need. Out of thousands of rare, hereditary, and acquired diseases, there are hundreds of disorders that are affecting 1 to thousands per year that could be addressed with novel gene therapies. And addressing molecular defects may reduce some more common diseases to very rare diseases. Actions at the Center for Biologics to address rare disease development include advancing manufacturing technologies for cell and gene therapy through research, applications of the platform technology provision, which will allow us to leverage information across products to expedite product development. To work to really clearly define the use of accelerated approval for gene therapies, exploring concurrent submission and product review with other regulatory authorities, and communication pilots for rare diseases. One particular effort in the Center for Biologics to highlight is our recent COGENT pilot, or the Collaboration on Gene Therapies Global Pilot, to help accelerate the development of gene therapies. Initial participation will be by standing regulatory members of ICH, and partners may participate in internal regulatory meetings and meetings that include the sponsors. Specific regulatory reviews are also shared across agencies and discussed with our partners. All meetings conducted and information shared is under strict confidentiality agreements, and the goal is really to increase the efficiency of the regulatory process, reducing time and cost for agencies and sponsors. The START pilot is an additional program that is used to further accelerate the development of rare disease products. And in particular, it is to accelerate the pace of development for products that are intended to address an unmet medical need as a treatment for a rare disease. There are 4 CBER programs that have been chosen and 3 CEDAR programs. The requirements for this program are that the product is being developed toward a marketing application. It's intended to address an unmet medical need as a treatment for a rare disease that's likely to lead to significant disability or death within the first decade of life. And with this program, you get enhanced communications with the agency and additional things such as ad hoc emails or live interactions on an as-needed basis. We are excited to highlight our START pilot selections in the Center for Biologics. We've chosen 4 products, as mentioned. Our first product is a recombinant adeno-associated virus serotype 9 vector encoding a codon-optimized full-length version of human NGLY1 for the treatment of NGLY1 deficiency. Our second product is an adeno-associated virus containing a self-complementary DNA payload of a codon-optimized human ASPAC DNA for Canavan disease. Our third product is a lipid nanoparticle encapsulated mRNA encoding a normal human protein, or mRNA 3705, for isolated methylmalonic acidemia due to complete or partial methylmalonyl coenzyme A mutase deficiency. And our fourth product is a recombinant serotype 9 adeno-associated viral vector, MECP2 transgene, for the treatment of Rett syndrome. The ARDEA pilot program is an additional program that seeks to advance rare disease drug development by providing a mechanism for sponsors to collaborate with FDA throughout the efficacy endpoint development process. Admitted sponsors may request up to 4 meetings with FDA to discuss development of their proposed novel endpoints. And after completion of 4 ARDEA meetings, the sponsor can request additional input from FDA as needed through other formal meeting mechanisms, such as Type B, Type C surrogate endpoints, or Type D meetings. This program is across both the Center for Biologics and the Center for Drugs. So in conclusion, I'd like to thank you for your time and attention. I've included several OTP resources here, which include guidance documents, our OTP events, meetings, and workshops webpage that lists upcoming events, and links to information and recordings of past OTP events. Interactions with the Office of Therapeutic Products website, which has a lot of information, and in addition, our OTP Learn webinar series. Thank you for your time and attention. And listed below is our contact information. Thank you again.